uh, uh, start. And so, well, hello everyone. And so then welcome to the Physics of Life South America Network seminars. And as you all probably know, the network is a venue to connect the researchers interested in physics of life within South America. And as you, well, most people at least seems to know, we have this three different thematic subnetworks: the ecology and evolution, cells and molecules, and neuroscience. And every month you have a seminar on each from one of these subnetworks. And and as you know, this network started as an initiative from of the ICTP to and started to foster this and to put us together. So and as you know, the ICTP-SAIFR is a kind of a branch from the ICTP, a collaborative effort that involves the Institute of Physics of Theoretical Physics from UNESP, and you find all the information on the web page. So I will encourage you to we have everything posted there, and you can sign for the mailing list. And and for our seminar today, it's a pleasure to have for our seminar Sergio Pantano. He's from the Institut Pasteur de Montevideo. Sergio is from Argentina, right? And so he got his bachelor's in physics in Universidad Nacional de San Luis. And then he got further studies at CISA in Trieste, and then he moved to a while ago already to uh, to uh, to Institut Pasteur, and he's a very well-known researcher in our field, and his research areas are on especially molecular simulations, coarse grain modeling, computational biophysics, and also design of biosensors. So today he's gonna talk to us about the challenges in simulating whole virus particles and some initiatives to address in the South. And Sergio, thank you so much for accepting our invitation. It's really a pleasure to have you. Please, the stage is yours.
Thank you very much, Victor, for invitation and also for the initiative. I think as we were just discussing, I think this is a very important part of the work. I mean, the initiative of linking the South American community. So, in that vein, what I would like to show you in the presentation today is something that we are doing in my group. There's going to be some propaganda here, but it's essentially what we have been doing in the group, developing some tools, and I will tell you during the presentation some of the context in the saying in the political or historical way in which we started with this stuff. But before starting, just remind me, Victor, that back in the time when I did my PhD at CISA, actually was nearby the ICTP building. And the ICTP was really fundamental to me because my PhD fellowship was paid by the ICTP. And now I am, I should also mention perhaps that I am a senior associate to the ICTP. So for the younger people, really do take a look. The institution is fantastic. I mean, the initiative, that, I mean, you can find a lot of programs there for fellowships and short-term visits and so on. But for today's presentation, I would like to show you what we have been doing in the group and with the idea of using coarse grain simulations to address virus whole virus particle simulations. In say the outline of the presentation is more or less what is written here in this recent review that we wrote with Luciana in my group. And so essentially the idea is that when we started to when I moved to Montevideo in 2007, 8 or so, we started to face some of the problems that we have in the south, that we don't really have a huge computational resources. And since the beginning, we were interested in some large systems, and we started to think that if we wanted to address those, then we will need some particular fixes that would make us competitive. So the field of whole virus simulation started in 2006 with a paper which contained this figure here by the group of Klaus Schulten. Was by the time was the first time that somebody simulated an entire biological entity. Was a satellite virus, the tobacco satellite virus with the RNA and everything. The simulation was really short, but was fantastic by the time. So it was 13 nanoseconds. The particle was not really stable even, but it really kind of started the field and started the foundational ideas that if you really want to understand the dynamics of the virus, then you really have to go for the big thing, because the protein-protein interactions or protein nucleic acid interactions that happens in the entire particle are not the same that will happen just in one single protein, because there is a lot of physics going on around that is not captured correctly in one single protein. The story of simulations have advanced, as you see in the right panel, with increasing number of systems and increasing the number of atoms contained in each of those systems. This is a portrait of a figure of a paper published in 2 or 3 years ago already, but it essentially contains more or less the progression of the sizes of the system being simulated by using molecular dynamic simulations. And as you can see, the number goes nowadays to 10 to the 8, 10 to the 9 million atoms. So this is the kind of challenges that computational virology poses to simulation, and it's something that is difficult to address, especially if you live in regions of the world in which computational resources are not so so widely spread as in the north of the world. So this should this is now an image from a paper by the group in just a few years ago already. And as you can see here, the number of publications, if you just look at the in the PubMed with the keywords by the viruses and simulations or molecular dynamic simulations, has been increasing in the time. The paper was published in 2021, and obviously, as you can see here, there was a peak in the number of papers during the pandemics. So there was a lot of interest and the scientific community in started simulating viruses. This where the viruses, and the picture hasn't changed very much in the last years about the kind of viruses that have been simulated. And essentially, as you see here, the size of the particles that you need to consider in order to have a realistic representation of the virus is quite high. And the details here are not very important, but if you see the size of the spheres here corresponds to the time simulated, and it goes to the order of the few microseconds in some cases. Especially for coarse grain representations. So essentially, again, this just reinforces the idea that trying to understand the dynamics of the viruses requires really, at least to begin with, a significant amount of computer resources. And what I mean with that is having the capacity to simulate systems for relatively long time scales, most likely in the order of the microseconds, and in size scales that are in the order or exceed actually the million of atoms. This is the case. I mean, one of the most recent ones is for SARS-CoV-2, but you can find in the literature and publications with all these kind of viruses. In the end of the presentation, I will just give you a glimpse of what we are doing with flaviviruses, which is the number 17 here. And this is more or less the landscape of what we will find in the field of simulation of whole viruses. As I was mentioning, when we started with these ideas, we immediately gained or bumped into the problem of computer power. If you go and see how the share of computer power distribution in the world, this is more or less a contemporary view of how the computer power is distributed. Most of it is it's distributed in China or Asia, but essentially China and Japan. So half of the computers in the top 500, it's distributed in Asia. Then the rest is in the United States. Relatively, I mean, still significant, but relatively small amount is in Europe. And just in one of these very small lines is what we have in South America. This is again from this review at the beginning of the year, so the picture is relatively recent. Obviously, Brazil is the country with more HPC facilities. The number here corresponds to the number of different HPC facilities, and as you can see here, there are not so many. I mean, there are some in Mexico. Sorry, I'm forgetting to say that these are for academic use mostly. So there are many other resources that are not listed here or not comprised in the figure, but these are for classes that are not for academic use. In particular, we have had the opportunity to use Santos Dumont thanks to collaborations with very good friends and colleagues in Brazil. And it's something that if you're not from Brazil and you're listening, it's something that is really fantastic. And some of the stuff I'm going to show you in the end, having done actually with simulations performed in Santos Dumont. So the point here is that you need to have the capacity to run simulations in relatively large facilities. But this is not the only problem that you will face if you try to describe the physics or the dynamics in a virus particle. Essentially, the point is that you are dealing with huge systems, several million of atoms, and usually when you run molecular dynamic simulations, what you do is actually you're not running one single particle, but actually you are putting your system in a periodic boundary conditions. But since the viruses are huge objects, they are usually resolved very poorly by experimental methods. Now the resolution is increasing and improving, but in general you will have a lot of problems that are related to missing atoms or missing residues, and this is essentially because viral proteins are slightly more complicated than proteins from eukaryotes or prokaryotes, and this is because the viruses have to pack the information in a very constricted space in the genome, and therefore the proteins have to evolve to be multifunctional. So essentially, it's very common to find in viruses proteins that are actually can have different faults. So they behave as IDP, so intrinsically destructured or disordered proteins. And this is usually a problem for obviously for structural determination. You will have a lot of loops or regions that cannot be resolved. This is also a problem because these loops are usually polybasic and are supposed to interact with the genome. And then when you take the cryo-EM map or the X-ray, I mean whatever is the experimental method you're dealing with, and you are using usually normally symmetry operations, all these poorly resolved regions in the protein will be averaged out, and eventually you lose the information. So essentially, this ends up in missing regions or missing atoms that you have to model. And of course, you can resort to stuff like AlphaFold or any other AI based resources. But the problem there is that they still remain a knowledge based method. So essentially, they will provide solutions in most of the cases based on structures that they have already seen. And obviously, if these are intrinsically disordered proteins, then there is no structural information. So um, usually, methods like AlphaFold will fail in the prediction of viral proteins. This is also the case because, for instance, the protein-protein interfaces in viral proteins are slightly different than the ones in mammalians, for instance. They are more rich in charged amino acids because they establish more salt bridges and exotic interactions that eventually can be more labile at low pH. So, and this is because viruses are engineered in such a way that they are metastable. So they have to be stable for some purposes, but then eventually they have to be also ready to release the genetic material, so they cannot be super stable. So this particular biological assemblies live in a sort of saying dangerous region in which the conformation is always compromised. This obviously also results eventually in long bonds or clashes because of the poor resolution. But on top of that, when you have to build up a particle, essentially what you know is a conformation of the proteins, most of the cases. So if your virus is, for instance, an enveloped one in which you have the genome and then a membrane coat, and then eventually some structural proteins, or proteins that are part of the envelope or of the capsid, then eventually you don't have the coordinates of the genome, or of course you don't have the coordinates of the lipids if they are. So essentially, what you will face is that you will need to learn some techniques which are ad hoc developed for that. And in that cases, eventually you can also end up in issues with density or clashes, or for instance, the incorrect saline concentration. There are many other problems associated with the protonation state that you have to assume for amino acids in the viruses. As I just said, many of these viruses live at different pH, and many of those have also what is called or known as a pH trigger. So essentially, if a virus like a flavivirus like Zika or dengue or one of those is in free in the solution, it has a mature form, then eventually it is incorporated into an endosome because it fuses, and then eventually it will go through, I mean, once in the endosome, it will go through and decrease in the pH, and this is what will trigger actually the exposure of some loops that will determine the fusion to the host genome. So all these processes are pH driven, and assigning the correct protonation state to particular amino acids is crucial usually for the stability. And it's crucial because in a virus you have several copies of the same protein. So if you make a mistake once, most likely it will be replicated that like even hundreds of times in the same copy of the same amino acid in different protomer which is present in the capsid or in the envelope. On top of that, if you use periodic boundary conditions, which is normally the case, you need to be sure that the sizes of the boxes are big enough in such a way that the interactions between the images are really neglectable, and eventually that if you will have some conformational issues like big changes in conformations, then you have to be sure that these conformers will not interact with their images in the neighboring cell. This is also related with the cutoff, so that how long you will consider the interactions. And of course, the bigger the cutoff, the higher the computational cost. So eventually, all these issues can sum up one on top of the other, ending up making the very setting up of the system already a really challenging task. For the simulations that I will show at the end, I mean, setting up a flavivirus, which contains in the order of 8,000 lipids and several millions of particles. In our hands, it takes like in the order of 3 weeks, 1 month, or something like that, just to have a well stable and converged particle with the correct number of lipids, water inside of the capsid, ions, and so on. So it's really something that is complicated. Um, One of the solutions that we found out eventually in time, and we are still using those, is the use or the development of coarse grain force field. It's our coarse grain force field is entirely and we are kind of proud of saying that it's entirely developed in South America and by South Americans. This is the team now. The force field is called Syrah, which is obviously just a complicated way of naming a wine. But it stands for South American Initiative for a Rapid and Accurate Hamiltonian. And the force field is completely integrated in Amber and is compatible with Amber. Actually, it's part of the official release in Amber since 2020. And it contains plug and play topologies and coordinates for essentially almost everything you will need to run simulations of biological systems. Everything but RNA, which is still quite complicated, not only for us, but RNA is really a complicated molecule. Just for you to have a glimpse, I will not go into the details of all the force field, but it's just something that is I always use because it's easy to understand. The coarse graining in our case is, and this is for the nucleotides on DNA, is more or less on this way. Essentially, what we use for coarse graining is the position of the real atoms that are placed on the coordinates of the atoms in the fully atomistic molecules. So as you see in sticks, is the heavy atoms, and then what you see when you have in semi-transparent spheres are the bits that make the coarse grain. So in general, for nucleotides, instead of having in the order of 30 atoms, we have 6 bits for each of the nucleotides. More or less the same for amino acids. We have 3 bits for the backbone, and then between 1 and 6 bits for the side chains of the bigger ones, like tryptophan. We have different representations for this for each amino acid. So essentially, the force field is aware of mutation, and you can really study the effect of mutations in the dynamics of the protein using these kind of simulations. And we have put a lot of effort in writing the force field in such a way that is written with the same Hamiltonian that you will use in many molecular dynamic simulations. So essentially, there is nothing weird here. We used essentially the same functional form, and we have terms for bonding and non-bonding. And in the non-bonding part, you have the usual Lennard-Jones or van der Waals and electrostatics. And as I said, the force field is now fully integrated in Amber and GROMACS, and eventually you can also use it in NAMD, because you can prepare the topologies in any of these two engines and then run simulations with NAMD, and we have also recently shown that. And it comes also with something we call Syrah tools. which is a bunch of TCL scripts and analysis for VMD, which is a very famous program in the molecular dynamics simulation community. And so essentially the thing or the workflow for using Syrah is essentially that you have to download the package, and this is from our web page. And then you just generate using a command line the mapping from all atoms to coarse grain. And then from that, you can build the topologies directly and run the simulations with Ember, Gromax, or NAMD. Just it's an important point here is a completely straightforward process, because Ember, Gromax, or NAMD actually don't know they are running coarse grain simulation, because they are just taking up a Hamiltonian, sorry, a force field that runs in the same Hamiltonian. So the MD engines are just running a normal simulation, and this means that you can do whatever you want that you can do in any of these engines. You can eventually do, I don't know, replica exchange or alchemical transformations or any kind of or whatever you can do and measure on a molecular dynamics. Eventually, you can do and measure with Syrah. Again, you have these tools for analyzing. This is how it looks like for a small peptide. You can start here from the fully atomistic version, map it to the coarse grain, use solvation ions, and you can set the ionic strength, and we have different kind of ions for that. And eventually you can even do the back mapping if you want, so you can recover after the simulation the coordinates for the fully atomistic resolution. And this is useful if you want to run some analysis on the trajectories that are not still included in our package. Very really just accepted. This was the cover was accepted yesterday. We have now a graphical user interface that will be helpful with the analysis. So this is going to be the next cover image in JSIM, and I will say a few words more if I made it with time by the end. So this is essentially what I wanted to tell you about the package that we use for running simulations. But coming back to the problem of setting up the system, even with the coarse grain representation, the situation is quite complicated because eventually you will need to put some pieces in the system that are not determined by the experimental methods. And for doing that, what we did was a very say fruitful cooperation with Leandro Martinez, who is a great colleague and also a good friend. And we contacted him, and essentially what we did was close collaboration to put together PackMol, which is one of the most famous programs to pack molecules and generate configurations for molecular dynamics, and put it together with Syrah to end up with a protocol that is optimized to build up molecular systems which are very highly confined. Essentially, what we did, this is in a paper that we published a few years ago, is to revise the penalty that PackMol assumed and once it was doing a Monte Carlo to place the molecules, and we ended up with the revised penalty functions that are better for placing lipids in particular regions as it is required, for instance, to construct particles of the Zika virus. Excuse me. So essentially, what we have now is a protocol that starting with the configurations that you have from the experiment is complemented using PackMol to place molecules in regions that are from which you don't have the coordinates, but you know, for instance, that the membranes are confined to some particular regions, as it is illustrated in this figure. This is the surface of the virus, and when you see in dashed lines are regions that are supposed to contain either the acidic tails of the lipids or the polar heads, because this is known at a very low resolution from the cryo-EM data, but we don't know the resolution of the atom. So we use PACMOL to place the lipids in the region that where they should be, and then we use Syrah to optimize the coordinates of that. Again, this was published in JCIM a few years ago. And of course, if you want to do that, or you are interested in running simulation of this virus or any other virus, I will be more than happy to help you with the setup. So in the rest of the presentation, I will just like to show you a glimpse of what we are doing with flaviviruses. Essentially, right now we have a couple of lines in the group dealing with the dynamics and structure of two families of viruses. One of this is the flaviviruses, and the other one is circoviruses. I'm not going to talk about circoviruses today, but just would like to, in the next 10 minutes or so, would like to give you some hints of what we are doing with flaviviruses. Unfortunately, flaviviruses are very well known in South America. They were not so well known like 20 years ago or so, or 15. But this is just what we need to know for the rest of the presentation. They are envelope viruses, meaning that they have the genome covered by a lipidic coat, and on the viral membrane there are proteins. The proteins enclose a positive single-stranded RNA genome, and their structure is very well determined. There are structures on many of these flaviviruses, not only Zika, but dengue, West Nile, Japanese encephalitis, tick-borne, and many others. So their structure, it's a very well determined icosahedral structure in the order of 50 nanometers in diameters. If you want to build up in a simulation box, you will need in the order of 20 or 25 million atoms to consider everything. And the other thing that is important, just for the sake of this presentation, is that the viruses contain, or the structure of the mature virus contain a fusion loop, which is very conserved in all flaviviruses. And they are called fusion loop because this eventually get exposed. They are not exposed in the mature particle, but eventually they get exposed during a low pH exposure, and this insert into the membrane and facilitate the fusion and eventually the release of the genome into the cell. So there are many, as I said, flaviviruses. They are transmitted by different vectors, and they are transmitted to different animals, humans among them, of course. And just for you to have a glimpse of the phylogenetics distribution, we have dengue and Zika, which are very closely related here. And these are belong to the family that is transmitted by mosquitoes, and they infect a lot of mammals and birds. And then there's this tick-borne encephalitis, which is obviously transmitted by ticks, and this can also, these are a serious problem mostly in Europe, in Northern Europe. And then there are others that are down here as Japanese encephalitis and West Nile, which are also neural tropism. So essentially what we can do is build up particles of that and run molecular dynamic simulations. What you see in blue now to the left is the cryo-EM map of Zika virus. And what you see in green and pink corresponds to the density that we calculated, the average density that we calculated during the simulation for the membrane and the proteins respectively. So essentially what you see here is that there is a very nice correspondence between what you will observe in the simulation after a few microseconds of simulation. This, I think, comes after 2 microseconds of simulation, So essentially, the process here is that we start the setup of the system with the coordinates only of the protein, and then we introduce the coordinates of the lipids. And then we introduce water in the middle. So all this region in the center or the core of the virus is supposed to be occupied with the genome, but actually we don't have the coordinates. So essentially, what we do, jumping some details, is that we fill that with water to avoid the compression by the external pressure, and of course added salt. It is sodium chloride in this case, just in the outside part. When you see and go to see, at least visually, the comparison between the blue and the pink and green regions is quite remarkable. And what you see and here in the lower panel to the left is the correlation between the cryo-EM and the simulations. Essentially, in the region that comprises the protein is super high, is in the order of 90% of correlation. So meaning that actually the density, the average density that you can obtain from the simulation is very close to the density that you can obtain from the experimental data. What happens with the membrane is a little bit different, because actually we started not with this kind of geometrical shape here, but with a spherical shape for the lipids. So they are distributed in a sphere, and this is what actually this very low correlation here in the green lines you see. So this is the perfect spheric system packed with PACMOL. But then very rapidly the simulation, without any kind of bias, go to increase the correlation, and it really finds the correct shape of the membrane. Actually, we see that the correlation here goes from nearly 0.1 to nearly 0.5. But this because we are not considering the transmembrane regions here. So eventually, if we would consider the transmembrane regions there, the correlation would be even better. However, what these pictures say is that we can run simulations and then we can reproduce the experimental data, which is great, but it's not very informative. I mean, this is just a kind of verification, or that we are doing the right things, but you cannot get many more features from this. So the point here, or the idea here, would be to use a simulation to extend our knowledge, or to try to get some new ideas or new insights about the dynamics of the virus. And in that regard, I will refer to this paper published a few years ago in Cell, in which they determined the structure of broadly neutralizing antibody called C10 that neutralizes both Zika and dengue, and actually different serotypes of dengue. There are 4 different serotypes of dengue viruses, and C10 is broadly neutralizes, so it neutralizes them all. In the cryo-EM structure published by the group of Felix Ray, this is what they determined. What you see here, if you focus for a second on the left, what you see in gray is the structure of the envelope of the protein, so it's the viral proteins, and what you see in yellow is corresponding to the electronic density coming from the antibodies that are attached to the surface. We will see that in a minute, but what you see here is that there is more density or more yellow coloring in these regions here than in this here and here and here and here. However, if you go now to the image more to the right, and you see now that the coloring is different, you see different grades of shades of gray, and each gray here now, it's a protein. Actually, there are 180 copies of the same protein. This protein is called protein E because of the E of envelope. And essentially what happens is that you have the same copy of the protein, which is repeated and forms in the mature form dimers that then associate into trimers of dimers that then generates a sort of flat patch on the surface of the membrane that then replicates over and over into the entire icosahedral forms. And so you have 30 copies of this flat patch that you see highlighted in green here. The point now is if you consider that this dark gray here is the same protein than this lighter gray here, then the region where this antibody is bound is identical to this region here where this second antibody is bound, but it's also identical to this region here, here, and there. But still, despite the fact that from the structural point of view, the binding site is identical because the proteins are supposed to be indistinguishable. Still, you have some preferential regions to bind, like that correspond essentially to the neighborhood of the three-fold symmetry axis. So we wanted to explore that thing and trying to understand why this effect happens. This is again is just a different representation, but what you see here is one protein here and one other protein here. This have a sort of L-shaped conformation. And what the idea here was running the simulation, analyze the dynamics, and focus essentially in some descriptor that we will indicate here with this dihedral angle. Essentially, what this dihedral angle measure is the angle determined by the center of mass of this blue part here, which is called as a domain 3. The red part here, which is the domain 1, and these two subdomains into neighboring proteins, which are called domain 2A and domain 2B. And essentially, what this dihedral angle here measures is how much these regions move. And eventually, the idea of why we would like to measure that is because this dihedral impact essentially in the conformation of the fusion loop. As you see here in the side view, the fusion loop is inserted and in principle not available from the exterior, which is this part here. I hope you are seeing my arrow. Yes? Are you seeing the...
Yes, we can see it.
Great, thanks. Sorry. So essentially, this fusion loop is not exposed normally to the surface. And this is because if the fusion loop is exposed, then eventually there is the risk that the protein will fuse to any membrane, not in the right time, not in the right place, not with the right cell, perhaps. And so the fusion loop is mostly exposed. But eventually, this differential binding of antibodies tells us that there might be some difference in the dynamics of the antibody. So we went to measure the exposure of the fusion loops in different conformations, and also the dynamics of this descriptor, which is essentially the dihedral angles defined by the center of mass of different subdomains in the protein. And of course, we measure this in all the possible symmetry axes. So we have these dihedral angles measured near the 3' axis. So there is one dihedral angle here, and obviously one here, and then there is another one here, and also near the twofold axis. So we can do that and analyze the dynamics. Before going into that, what we saw in the simulations, and now what I am showing here in the right is the dynamics. In the bottom panel is the exposure of this loop during the simulation, not only for one flavivirus, but this is for Zika, dengue, Japanese encephalitis, West Nile, and tick-borne. As you may remember from a few slides before, these 5 viruses are distributed more or less in all the phylogenetic tree of the flaviviruses. So we suppose they are enough to be representative of the diversity that we will have in the flavivirus family. Actually, strictly speaking, it's flavivirus genus. And this is what you see. If you analyze the exposure of the loop as a medium, as an average, you will find that depending on the virus and depending on the symmetry axis, you will find different values. Zika is much more stable, and we know that already from simulation. So Zika is even more thermostable as a virus. And so when you go to dengue and much more to Japanese encephalitis, which is the most labile flavivirus in this set, actually it is so labile that unlike the other ones, this dimer is not actually stable in solution in Japanese encephalitis. It only is stable in the presence of the entire particle. So it's not surprising that the exposure changes a lot. And as you've seen, it not only changes with the virus, but also changes with the symmetry point in which you measure that. And this is what you see here in the bottom line of images is the trajectory of one single of these fusion loops that you have 180 copies in all the particle. And what you see, and this is just because just as a representative image, is that these viruses, different viruses, have dynamics that are in the order of the microsecond time, and then eventually what you see is the exposure goes up and down, meaning that the fusion loop goes up and get exposed, perhaps in a sort of sensing mechanism. So the loop is exposed, see if there is something interesting to fuse, and if not, it goes back down to a low exposure region, in which is very similar to this conformation here, in which is contacting the domain tree. Then, if we go to analyze the dynamics of the dihedral angle, and this is one of my last slides. This is what happened. Now I'm restricting because of the time. I'm restricting the illustration just to Zika and dengue, but in close correlation with what we see here with lower exposure. When you analyze the trajectory of the dihedral angles, you also see, and this is what you're seeing here. This is the angle, and this is the simulation time for Zika on the left and dengue on the central part of the figure. When you see these darker colors, means that you have more dihedral angles populating that region. And what you see in dashed line is the angle that you will measure during the experiment. So essentially, depending on the symmetry axis, you have a different, not only a different average value, but also a different mobility in the loops. In the dihedral angles that is correlated with the exposure of the loop. So essentially, the threefold axis is more mobile in this Zika, but there are some differential behaviors between Zika and dengue. So in this region here, in the twofold axis, dengue is much more wide than if you compare with the results of Zika. We did the same thing for all the flaviviruses, and this is what I am showing here in this panel here. This is a twofold comparison between the average value, so the position of the peak or the most populated region in the dihedral value and the dihedral angle, comparing thick-borne West Nile, Japanese encephalitis, dengue virus, and Zika. And what you see in colors correspond to a definition that is arbitrary, but essentially, I'm sorry, there's a construction in upstairs, so I'm sorry for the noise. What you see in green is because, or it means that West Nile and thick-borne encephalitis have a very high correlation in the twofold axis. So essentially, the green means that there is only 1 degree in difference in the angles, in the maximum, sorry, not in the maximum, but in the average value. When you see red, is because there are larger differences in the positions of the average values of the angles. And the same you can do for twofold, threefold, and fivefold axis. And essentially, what you will find in the end is something that is kind of quite interesting to us is that you can accommodate it with using this dynamic descriptor that will reproduce the same reorganization that you will have with the phylogenetic tree. I'm just realizing there is some Spanish there, but okay, it's pretty much the same. This is what you have with phylogenetic tree. If you order the flaviviruses, this is more or less what we saw before, and this is what you will get if you generate a tree that is based on the dynamic description that you will obtain from the dynamics of this dihedral angle. So essentially what happens, or what seems to happen here, is that there is a correlation that goes from the say 1D structure, so only the genome of the virus or the one of the primary sequence of the proteins, that then translated into the 4D picture, in which not only the structure is similar, but also the dynamics that results from that structure is similar, at least in what regards to the dynamics of this region that comprises the fusion loop. So essentially, this seems to suggest that the recognition of which is preferential in the regions of the fusion loop that is most populated in the threefold axis is associated or is highly correlated not only to the structure of the virus, but more to the dynamics of the virus, and this is also correlated in time with the 1D structure or the sequence of the genome. So there's a sort of unseen yet correlation between the sequence, the structure, and also the dynamic that it determines. We think that this is something which is key in the recognition. So this is why, for instance, there is this effect of cross recognition between antibodies between dengue and Zika, which is not only related to the static structure of the viruses, because actually the antibodies recognize regions that are not similar in the surface of the viruses, but they're similar in the dynamics of the viruses. And the dynamics is related to the icosahedral structure, which is conserved, but also to the dynamics of the small and more flexible regions of the virus, which corresponds to this fusion loop. I think with that, I will just stop because my time is running up. And we'll just let you with some conclusions. Essentially, in what regards to the difficulties of setting up topology of viral particles, we have already something that comprise a pipeline for the construction of stuff, which I will also would like to point out that is entirely South American. I mean, it's made as a fruit of a cooperation between University of Campinas and the Institut Pasteur de Montevideo. This, when combined with a force field which is coarse grain, and so in the order of 100 times faster, depending on the engine you use, can give you the possibility to run simulations of entire virus particles that you can actually run for several microseconds and compare with different viruses. So it's something that is really attainable to us in the present time, not only for flaviviruses, but as I said, we are also working with other viruses. In the specific focus of flaviviruses, it seems that the conformational dynamics is symmetry related. So this icosahedral shape that is conserved in many viruses is really intimately related with the antibody recognition, but also with the similarity that viruses have, not only in the structure, but starting from the primary structure or from the sequence, and it propagates to the 4D. With that, I will stop, and thank you very much for your time, and I will be happy to answer questions. But just let me go very rapidly to another stuff I would like to drive your attention, which is here. There is an open initiative for and I think it's pretty much in the in the lane of what we were discussing at the beginning. There is a virtual issue for computational chemistry in the global south. Essentially, this is just dedicated to authors from Latin America. You can submit papers to JSIM or JCTC. The virtual issue is open until October. This October this year. So if you are considering submitting simulation or papers with related to computational chemistry or physics to any journal, please consider to do that, because it's again it's an initiative that started also from friends and colleagues. In particular, Teresa Soares is the driving force here, who is also a good friend. And if you are considering submitting a paper, please consider to submit a paper to any of these two journals. Will be fantastic to have a very large number of papers showcasing the production of Latin America in this area of science. And with that, I will stop sharing and hopefully answer questions or comments. Thank you very much.
Thank you so much, Sergio. Fantastic talk. Just amazing. I wrote a lot of questions along the way, and as I was writing them down, you were on the next slide, so you were just answering them. So it was very nice, very
Good.
pedagogical type of lecture. So, but before I don't want to start with questions, so I hope people have. If people, I will open for discussions, for answering questions. So Sergio.
Yeah, hello. Hello. Thank you very much, Sergio. Very nice presentation.
Thank you.
Question about the Sierra force field.
Sure.
Here you a system which is a very big one, like the virus, right? How it works with a smaller system, like for instance, the interaction of a lipid with a membrane. Just, I don't know, because the lipid is very small, but
Yeah.
it's challenging for MD also. So for pure all atom MD, right? How do you think the Sierra force field will work there?
I mean, membrane simulations are complicated per se, not just coarse grain. But I think in the particular case of coarse grain, membrane simulation is something in which they do well. I mean, Martini, which is the most famous force field at the coarse grain level, started with lipids, and they do a great job. And I also would say that I mean, any other force field works well with membranes. And in our case, perhaps the limitation is the number of lipids that we have. So we have currently in the order of 30 different lipids. We are preparing a paper. We are writing that, and we are expanding the diversity to 100 nearly of different possible lipids. But in principle, you can. I mean, we have a few papers published, and from what we have seen, and other people also, the dynamics of the membrane is quite well reproduced. Even in our case, we can reproduce the preferential sampling of lipids around specific places of proteins.
Sorry, about the interaction with the membrane with
yes, I mean the specific interactions, protein membrane interactions, like I don't know, arginines that I'm going in some technicalities. I mean, the tryptophans do something that is called floating, and arginines do something that is called anchoring, because they go deep into the membrane and interact with the glycerols. And lysines do something that's called snorkeling, because they are in the membrane phase and they go up, and all these kind of things are well reproduced in our case.
Thank you a lot.
Yeah, in principle, we are happy with the membrane part, and the problem for us until now was the diversity, but we are just about to submit a paper including cholesterol and ceramides and sphingolipids and so on.
Thank you a lot.
But if you have any system, just send us an email and we'll be happy to help you.
Right. Thank you.
Anyone else wants to ask questions? So, well, while nobody says anything, I have a few questions. So, Sergio, if you have like two situations and with a docking something, some potential good drug, do you see, if you run with those things, have you tried to include those? Do you see any significant changes? And the question is, when you do you have like good reaction coordinates that you can see this switching to have like big transitions from some like one basing to another? How do you probe this? What would be this excited states? Could you comment on that?
I should start by saying that in general, doing docking, small compounds and coarse grain may not be a good idea because you are losing details with the coarse grain. So the specificity in the binding most likely will be ruined by the coarse graining process itself. And it's not only in our case, I would say in general. Still, we have seen and some interesting cases in which, for instance, we have a FRET sensor in which we have a module that senses a compound, and when the compound is present, there are two fluorescent proteins that remain stable or relatively close one to each other, so they can transfer energy. And then when you remove the compound, then the two fluorescent proteins go far apart one from the other, and then decrease the FRET signal. This kind of stuff is very well reproduced in our system. We are working with some people that ask us for some specific compounds to do the parameterization. We don't have any recipe now or so far for producing small molecules. So it's kind of custom made, but in principle, yes, we have seen and we have a few papers published with these kind of things in which proteins and essentially what you see is an allosteric trigger. So essentially, when you put the compound, then the protein remains stable, and then when you remove the compound, then the protein goes into a different state. I don't know if this addresses your
yeah, like for example, these kind of changes, such as, for example, if you look at different lineage of the same virus, for example, do you have sensibility to see why, for example, you have some lineage that is more aggressive, or have you probed these type of questions? You probably are thinking this is on the line of the things that we plan to do in the future, trying to look at the different trajectories and see how we could think about things.
I see that Martin Soñora is in the audience, and what he's doing right now is introducing mutations in Zika that comes from the different strains, and they have different aggressivity or infectivities. So we are trying to analyze the effect of the mutations and trying to correlate with one of these descriptors. If we can see something that difference and can be correlated, because eventually the idea would be to generate mutations that attenuate the virus, so they cannot be so ready to fuse. So if you could generate the virus which is very stable, then you will increase the chances for antibodies to recognize you will increase the chances for the immune system to recognize the mature particle rather than an open how to say, you will recognize the mature particle, and that will eventually generate neutralizing antibodies. which would be preferable to a situation in which you have the broken isolated proteins that will generate an antibody response that will be not neutralizing. So we are working with that and introducing and comparing actually mutations from different strains to see if we can correlate that with the dynamics of the virus.
There is a question in the chat, Vitor. I don't know if you want to take it.
Let's see. Oh yeah, let's see the.
Yeah, it's by Vignesh.
Yes, yes. Yeah, we have a question here. And
okay. No, we don't have, I mean, the question is, how do we, in many force fields, just let me give you some context. Many coarse-grained force fields use an elastic network to hold the structure, because since they lose some details, eventually the proteins will just fall apart. In our case, we don't use any elastic network. So these fluctuations in the loop that you see, they are spontaneous, and these are triggered just by thermodynamics. I mean, the more or less flexibility of the loop in the particle is determined by the sequence itself and by the structure itself. It's not related to any elastic network, so we don't use this kind of tricks to maintain the structure. In that sense, Syrah, it's a force field just as any other that you will use in fully atomistic molecular dynamics. So the conformational, you can have conformational transitions, and you can lose or gain secondary structure elements, helices or beta strands, or these kind of things. This will happen spontaneously. The force field is not good for folding, but it can recognize when it has to open or close. I think this is what.
do you on those lines, do you use like structure-based models, like
No.
potential?
No, the force field is parameterized against structures. So we use, I mean, the alpha helices and beta strands are in regions that correspond to in the Ramachandran plot to the more favored secondary structure elements. But then we don't use. I mean, this is only used in the stage of parameterization. Once we have the parameters, then the simulation runs just as any molecular dynamics simulation.
I see.
So there is no elastic network at all.
Oh, any other question?
I have a quick question, perhaps. First of all, thank you very much, Sergio. How do you see the progress of the field? And at the very beginning, you showed this graph showing how things have evolved in the past years. How do you see this for the future? Which kind of revolutionary things we can do, and when, in a couple of, I don't know, 10 or 20 years?
You mean when you see the field, you mean the coarse grain or computational virology?
Computational virology, yeah.
Computational, okay. I think, I mean, when the beginning of AI based methods, the thing will explode, and now we are seeing more and more use of coarse grain methodologies to run the dynamics of entire virus-like particles. Actually, and I mean, you can see that when you go to conferences on physical virology, that the people is looking more and more for these kind of approaches, because there are many things that cannot or will not be solved in the near future by experimental techniques. I mean, the problem of the genome inside the virus, it's very unlikely to be solved, because when you do the symmetrization, you can fit on the proteins because the protein repeat themselves many times, and so it's always the same. So one rotation will give you the same protein, but when you introduce that rotation, then obviously the membrane, but also the genome inside, it's completely blurred because you're averaging things that should not be averaged. So this is something that would be for sure the next frontier for computational virology. Now we have the capacity to run simulations either at the coarse grain, but I mean, people in the US or Europe or Japan, they can really have the computational resources to run big simulations. I mean, we saw that during the pandemics, all the story about the glycan shield and how the sugars move around the spike protein in SARS-CoV-2 is something that could only be solved by molecular dynamic simulation or by computational stuff, because you cannot see that in by experimental methods, because the glycans are heterogeneous. So you have the same proteins, but you have different glycoforms. And this is something that is relatively easy to study with computational methods and very complicated to study with the structures. So in the regions where the proteins are very well resolved or determined, that will go to cryo-EM and all these new, relatively new methods for cryotomography and everything. But then there are many other things that are not solved and even not solvable, at least in the near future. And I think that the computational part has to address that points. I mean, something I didn't mention, but Zika or flaviviruses are glycosylated, and the role of the glycosylation is not very clear. And the reason it's not very clear is because we don't have tools to study that. Even because the glycans are not the same in any protein, so the heterogeneity it's complicated. So I think in the near future, this is going to be, I think things are going to evolve more, to include more parameters and techniques that will allow to have a very match or complementarity with the between computational and experimental structures. This is my opinion.
Exciting times. Thank you.
Yeah, well, the good news from what you were saying is that the most important part in terms of trying to come up with drugs, etc., lies on looking at the surface, on the membrane. So that still is good news, because whatever is inside it was going to be a different story, right? The most important thing is how the interaction with other systems takes place. So
absolutely. And then also the multi-scale part, which I didn't really mention, I think is important, because in some places you need to have some really detailed interactions, but in other places you don't. And we are also trying to move into that direction, and having some regions of the system at the fully atomistic resolution and some regions at the system at the coarse grain level or supra coarse grain level. This is also something that will accelerate the simulations, and it's something that is moving fast. Also because now with artificial intelligence, it's easier to understand how one part of the system may communicate with the other, because if you can learn that from simulations you already have, then the interaction parameters between atomistic and coarse grain could be easier to develop than there were a few years ago.
I have a question, because you're mentioning AI applied to, getting us input, learning from simulations, basically, not from data, from experimental data. And you mentioned that it's very hard to get some data with the level of detail that you get from the simulations. So my question is twofold. On one hand, how do you validate your simulations if you cannot contrast it with experimental observations? And if you use the simulations to feed the machine learning techniques, aren't you going into a loop in which you have a bias that comes from the simulations themselves, and you don't have the input from the actual observations? How do you solve that?
Yeah, this is a super important point. And indeed, there are people making those mistakes. In our case, we compare only, during the parameterization phase, obviously we have a lot of controls, and we only compare against experiments, and not only 3D results like X-ray or cryo-EM or whatever, but usually also with in the case of dynamics, we compare against fluorescence data, or I don't know, if you have interactions data, then whatever it is or NMR or this kind of stuff. In general, we don't compare against simulations because, in principle, well, on one hand, there is a risk of what you mentioned. On the other hand, it's just comparing one model with another model. The other model could be better, but for sure, we should compare against experiments. And then we, as usually, we extrapolate. I mean, in the case of we are something I didn't mention. I mean, the other study we are working on right now is to put in the genome inside of a small virus and to understand the contact regions and everything. But in every case, you just go and check that, for instance, the interactions are those that are known from all the family, or you use the computational part not only to study the structural dynamics, but also to check against, for instance, alignments in the sequences, so that you know if you have certain variations in a virus, then you know that these variants are allowed in nature. So eventually those interactions may or may not be important. So you can kind of extrapolate your results and compare against not only structures, but also, for instance, statistic information that comes from the variation of viruses. And so but this is a complicated thing, and yeah, the possibility of remaining in a loop is something that you have to keep in mind at all the times. Yeah.
Thank you.
Well, thank you so much, Sergio. It was a fantastic talk.
Thank you.
I congratulate you not only for this talk, also for the initiative on the Global South with Teresa Soares. Is a very nice thing, pushing exactly our community on these directions. With that, I think we had a fantastic seminar. I don't know if you want to have some final words, Sergio.
No, just looking forward to the next seminar. Thank you, and I guess we will meet in a month or so.
Yes, hopefully we will announce it soon.
Great.
We will. Well,
we
with that, with that, I thank you so much. Thank you for all that came, and and see you in the next seminar. Thank you all.
Bye. Thank you.
Bye.
Okay. Bye-bye.